All right, what a story. Yeah, Laura, thank you and everybody at the foundation for everything that you do for both the patients and the basic scientists. We're really grateful, particularly during this challenging times. Okay, so we're in for a real treat because we've got one of the world's leading neuroscientists with us, and I don't say that lightly, in Dr. Don Cleveland, who is a distinguished professor of cellular molecular biology at the University of California in San Diego. So, Don's discoveries have really transformed our understanding of ALS and more broadly neurodegenerative diseases. He has made a number of seminal discoveries over the last 30 years in ALS. And beyond, to name a few. He was the first one that discovered tau back in the late 70s. He went on to uncover how neurofilaments can cause motor neuron death, which I think is what initially triggered his interest in this terrible disease. He was instrumental in thinking beyond the motor neuron in the context of ALS through a series of beautiful transgenic mouse model papers. He showed that there's non-cell autonomous effects in disease. More recently, he brought to our attention the importance of loss of function effects downstream of TDP-43 in identifying staffmin, which is an exciting therapeutic target. And perhaps most notably, Don was instrumental in the development of antisense oligonucleotide approaches for ALS, specifically for SOD1, which has really transformed the field. And it has transformed the field because it proved to us that we can actually treat an ALS patient if we get the science right, if we understand what is the cause, start from genetics, if you understand what are the mechanisms, you design a rational therapeutic, you can make a meaningful difference. Now that development has gone beyond, obviously, the implications in ALS, because antisense oligos are exciting treatments across a number of different neurodegenerative diseases. So I will say that obviously he's a member of the National Academy of Sciences, National Academy of Medicine. He's won a number of awards, including the so-called Oscar of Science, the Breakthrough in Life, which was an incredible achievement for Don, but it also reflected on the entire community. But I would say that his legacy goes beyond his scientific achievements. I think one of the most important parts of his legacy is all the trainees that he's put out there. And, I can name 10 people off the top of my head that came out of Don's lab. Some of them are in this room today that have gone on to become leaders in the field, and they are in critical positions. So I think that's a pretty substantial part of Don's legacy. And the last thing I want to say is something that hopefully he will appreciate, which is a couple of years ago, after I think a Target ALS meeting, a bunch of us young sort of mid-stage investigators were out at a bar after a meeting and talking about ALS and the state of the field. And somebody asked, well, who do you think has been the most important researcher in terms of ALS over the last years? And, there was only one answer to that question. So if I can borrow a term from the sports industry, we all thought that Don is the MVP of ALS research over the last few years. And, we're grateful you're here with us today and looking forward to an exciting next hour. So, Don, thank you for being here.
Well, thank you for that all too generous introduction. It's always great to be back here in Chicago at Northwestern. I remember turning down an opportunity to join Northwestern for my first job. Yes. Anyway, so what I'd like to do today is cover some of the points that Evangelos had just raised in the step moving forward from the discovery of the first cause of ALS to the first demonstration of an effective therapy for an inherited form. And the story actually really began. Yeah, the story really began with the recognition that from the genetics of human neurodegenerative diseases, that all of the big diseases, Alzheimer's, Parkinson's, ALS, Huntington's, where we know that there's a gene or genes that are contributory, causative or contributory to disease, they're widely expressed in the nervous system. They're not expressed uniquely in individual classes of neurons whose dysfunction gives rise to the disease. But even more so, it was the discovery initially by Andy Singleton, then at the NIH, that if you're supposed to inherit one copy of each gene from mom and one from dad, but if you happen to inherit 3 copies of one gene by a mistake during a phase called meiosis, you end up with 3 copies of a perfectly normal gene, and that gene encodes a product called alpha-synuclein. You will develop early onset Parkinson's disease. Too much of a good thing. And that was then followed by a similar discovery that if you inherit 3 copies of a completely normal gene encoding amyloid precursor protein, you will develop early onset Alzheimer's disease. So in those instances, you could see that a completely on-disease mechanism therapy would try to reset synthesis back to the normal level. So 150% extra is enough to be causative. And the disease we started with was ALS. Of course, all of you in the room know that. It's a selective death of upper and lower motor neurons. Almost invariably fatal over a handful of years. Your lifetime risk is somewhere between 1 in 1,000, 1 in 2,000. And biologists are not really that good at numbers. You can just take the world's population, and that means that there are millions of people now alive who will die from this disease. But the beginning of the scientific discovery was that about 10% of instances are clearly inherited. And I should have said right at the outset, I know that there's an online audience. I know that there's some patients and caregivers. There's going to be some hardcore science, but there's also going to be content that's available for absolutely everybody. So if you get tired of the science, don't worry. We're coming back to the things that everybody can grasp in a moment. I have to put some of the science in, otherwise my colleagues here will think that we're not really doing science anymore. Okay, so here's the founding discovery from investigators worldwide, including Tipu Siddiq, who was here at Northwestern. In investigators worldwide, neuroscientists and geneticists discovered that mutations in a gene encoding something called superoxide dismutase was causative of about 20% of inherited ALS, which is about 2% of overall ALS. And there's an article, a Newsy article accompanying the original publication, proposed that these mutations, they damage a gene and that they would lose. The gene would not be able to encode a fully functional gene product. It would be loss of activity. And it said right there, did radicals strike Lou Gehrig, meaning this enzyme is able to take oxygen with an extra electron, superoxide, and convert it into something less dangerous. And we now know that there are mutations all over the structure of this protein. Here I drew this now 10 years ago when there were about 140 mutations known. There are even more now. And even more so, we made, right at the beginning, there were 11 different mutations identified in this founding work. And we made 6 of them and showed that one of the mutations retained full activity as a dismutase. Completely. So specific activity was the same as the wild type protein, and another was totally inactive by every assay. And then the others were in the middle. And then we took I took mice and made mice expressing the fully active mutant and the fully inactive mutant, and asked, do either of those cause fatal paralytic disease in mice from expression of that human-linked gene mutant? And the answer is yes, they both did. So no activity, 100% activity. They both cause disease. And we took the one, actually Lucy Bruin, many in the audience know she became the first scientific director of the ALS Association. She's well known around the world for her contributions there. She took that mouse, she characterized the mouse that develops disease despite having two endogenous genes. These mice have normal levels of dismutase activity, and an inactive mutant, and they develop fatal motor neuron disease. And then she deleted one of the endogenous genes, and then she deleted both of them. So she had mice with an inactive mutant that had normal, half, or zero dismutase activity. And you don't have to pay much attention to which color is which. The presence or absence of dismutase activity did nothing to disease progression. So at least for disease in rodents from an ALS-linked mutation in superoxide dismutase, dismutase activity does not matter at all. Okay, and actually there was a contentious time where many in the community still thought, well, no, it's going to be reduced activity. It's going to be reduced activity. And actually now after that contentious time, we have settled as a community on there's one or more toxic properties of the mutant protein. In 2025, we still don't agree on what's the most important toxicity. And I'm not going to cover those today, but my own personal view is that everybody's a little bit right, that there are multiple pathways through which the mutant protein can disrupt normal neuronal function. But even more so, and thanks to Evangelos in the introduction, even more so, this gene is expressed in every cell in the body. So is it true that a red neuron, a mutant-expressing neuron, synthesizes the protein that damages it, and if it's surrounded by normal cells? Or is it actually the other way around? Would a normal cell acquire damage from a surrounding mutant-expressing neurons? And we thought, let's test that. And of course, neurons live in neighborhoods. They're connected to many different kinds of cells. There was a feeling that it had to be the neurons. Well, we don't have as many old timers in the audience. I could pick, if I name somebody, they'll be insulted. And if I don't name them, they'll be insulted. There was a very strong feeling, actually, that it was really the neurons that mattered. It was synthesis in the neurons. And we tested that initially by making chimeric mice that were mixtures of normal cells and mutant cells expressing the mutant at high levels. And when we did that, we happened to make mice that had 100% of their motor neurons were mutant. And we knew that because we genetically marked them so the normal ones would be red and the mutant expressing ones would be green, and we could count them. And we ended up making mice that had 100% of their motor neurons were mutant, expressing mutant protein at high levels, and none of them were lost. They did not develop disease. At least they didn't develop disease in the time course that we allowed them to live. This example was 35% mutant, but we had some were 100%. So it couldn't be true that synthesis of the mutant protein just in the motor neurons really mattered. And so then we tested, so which cells did matter? And so we built one of the first examples of a gene for the scientists where we could put the gene in every cell, but then activate the gene only in selected cell types by using a Krelok's approach, something that's now been used many, many times. And in our founding example, we used that to remove the mutant protein from the motor neurons, and that strongly delays disease onset. Okay, so synthesis in the motor neurons matters. But even more provocatively, if you targeted to remove mutant synthesis from the glial cells, the supporting cells of the motor neurons, the first one, the innate immune cells of the spinal cord, you don't change the timing of disease onset by lowering mutant synthesis in microglia, but you triple survival after onset, something that's never been achieved by any standard in a therapeutic in mice or in men. And so the microglia do something when they synthesize mutant protein that drives rapid disease progression. That was also true for a second glial cell type, the astrocytes, and it was true for the oligodendrocytes. So the three major glial cell types contribute to driving rapid disease advance. It's something that really, if you're going to make a therapeutic approach, maybe you want to target the cells who are driving rapid disease advance. And some cells didn't matter. So the muscle cells did not matter. Okay, so the endothelial cells didn't matter. And that was met with a highly contentious time as well. And I have to say, to give Northwestern their credit, one of the major opponents of the idea that cells other than motor neurons could drive disease was at Northwestern. And there was a very contentious time. And that reminded me of this German philosopher in the mid to late 1800s, Schopenhauer, who said, all truth, and I think he meant new truth, passes through three stages. And first, it's ridiculed. Yep, that happened. And then the phase two, for those who've been around in science, you'll know this is true, but to me it was counterintuitive. The second phase is the hostility gets even stronger. And then you get to the third phase, where everybody recognized that it was obvious in the first place, and that we always knew it was true. And that's where we are today. So we know the disease is non-cell autonomous. It's from damage from mutant synthesis in a variety of cell types, one of which is the motor neuron, whose dysfunction drives the fatal paralysis. Okay, but in any case, we thought, well, okay, can we just turn the bad gene off, since we know it encodes something bad? And we set out to try to do that. And the strategy, for the young scientists here, if I said, how will you do that? I know what you're going to say. You're going to say, use an siRNA, but no. We're going to use an older strategy of taking what I call a designer DNA drug, a little snippet of DNA, the substance of genes, and if you can get it single-stranded, if you can get it taken up by the target cell, it can diffuse into the target cell nucleus and base pair with the target RNA, a pre-mRNA. And that makes the RNA, but not the DNA, the DNA drug, a substrate for an enzyme that's present inside the nuclei of almost every one of our cells, RNase H. And RNase H's job, at least one of its jobs, is to look for things that look like viral things. And it cleaves, and as part of its job is to cleave the RNA, destroy the RNA in an RNA-DNA hybrid. So we're going to use that. It's a very simple mechanism. If you can get the drug into the right cells, you're going to get catalytic destruction of the information intermediate from the gene to its final product, the protein. Okay, and everybody knew this could not work. And the breakthrough was we did it anyway. These drugs are. A typical drug is 500 Daltons in size. These drugs are much, much bigger, 7,500 Daltons. They're heavily negatively charged. There was no known mechanism to get these things distributed broadly in the nervous system and taken up broadly across the nervous system. And yet when we did that, it actually works pretty dang good. And I admit that there was deep skepticism everywhere, including from me. And I was hassled by a local neurologist, Richard Smith. Some of you will know Richard, and he was adamant that we try it. And we actually showed. I'm going to show the outcome of experiment 1. If it hadn't worked on experiment 1, there would not have been experiment 2. because it was sure to fail. And over the years, the medicinal chemists, for the scientists, have modified the phosphates and to make them thiophosphates, and modified the 2' and 5' positions of the ribose sugar, and to make ASOs now that have very remarkably long efficacy periods. And so the best of them, you know, when you take an aspirin, you can get, you know, 3 or 4 hours worth of relief. And it astounded us to see that with these class of drugs, we didn't get 3 or 4 hours. We didn't get 3 or 4 days. We didn't get 3 or 4 weeks. We got 3 or 4 months worth of efficacy from the best of them. And so, we thought we were going to have to continuously infuse these into the nervous system. And we went to the pump maker, Medtronic, that does infusions in the nervous system. And it was very clear that there was just a meltdown and we thought the project was dead, because the Medtronic was insisting on owning 90% of the drug. And that was just a no-go for a company that never had a... I forgot to mention on slide 1, I've been partnered in this adventure with Ionis Pharmaceuticals from the beginning. And for Ionis, there was just Medtronic's terms were just completely impossible. And then I got the big team together, 3 of us, and somebody said, well, why don't we just inject it in and see how long it lasts? And we did that. And we learned that we could get, in that era, we could get 2 to 3 months worth of efficacy from single dose injections. And here is the founding experiment. We're looking at the level of the RNA for SOD1, and this is a transgenic rat that has a very aggressive disease course from expressing an ALS-linked SOD1 mutation. And we could see, here's the level of the RNA in control animals, and here's the level, and we could suppress that level of RNA near the site of infusion of our drug. We're injecting the drug into cerebral spinal fluid that bathes the brain and the spinal cord. And farther away, we could see suppression. We could actually see suppression throughout the entirety of the rodent nervous system with a focal injection into cerebral spinal fluid. We then very quickly showed that was also true in non-human primates. And now we dosed these, we dosed these animals at disease onset. And we asked, well, so what happens? And I told you that these animals have a hyper-aggressive paralytic disease course where from the time of phenotypic onset till we have to euthanize them is only 25 days in the saline-treated animals, but in the founding experiment, we doubled survival after onset by suppressing SOD1 dosing at onset. So we thought we'd really done something. And so we sent that for publication. And we sent it to Nature Medicine, and here's what came back. It was editorially rejected. It was rejected because there was no conceptual advance, because we had already demonstrated that there was a toxic property from the mutant protein. So if you could turn the dang thing off, of course that would work. And I got the editor-in-chief to take a second look. And he wrote, well, it's a nice demonstration of the effectiveness of the modified oligonucleotides. And in the founding paper, we had demonstrated the utility in non-human primates. And he confesses that this experiment of non-human primates was remarkable. But it wouldn't be therapeutic because it would have to be injected to be effective. And I said, I thought we injected things in medicine. And he said, it will never be the basis for human therapy. Okay, and I mentioned that for the young folks, that if you think that the one trait that you're going to need in your careers, smart is good. It's neither necessary, super smart, neither necessary nor sufficient. Great experimentalist, neither necessary nor sufficient. Okay, there's a third one. It's not sufficient either, but it's absolutely necessary, which is perseverance. Because you're going to get, I've got a whole drawer full of these rejections, sometimes from the work I know is the best of the work that we've done, and yet that's what you see. So you have to have perseverance, and there was the founding paper. We did realize that it could be broadly useful well beyond just SOD1 mediated ALS. It could be broadly useful in neurodegenerative disease. Okay, so now, if you fast forward from the initial work was done in 2003, 2004, and now if we fast forward to 2022, the phase 3 trial to use an antisense oligonucleotide to suppress SOD1 synthesis in ALS patients was completed on June the 3rd, 2022, where the green curve shows loss of muscle strength in the treated patients, who did better than if you look at the blue dashed curve, than the placebo-controlled patients. So they did better, but disappointingly, they didn't quite do as well as we had hoped, because the placebo patients did much, much better than expected. And that, I think, was because there was hope. There was hope that it could be effective, and we got people much earlier in disease in the trial altogether, and therefore it made the statistical significance harder to see at the 6-month time point. But then all patients were allowed to take the drug. The formal trial was over after 6 months. But now in the open label extension, look what happened to the treated patients. So in the second 6 months, 25% of them clearly got better. So we didn't just slow disease progression, we got partial disease reversal. And that's absolutely, at ALS meetings today, you'll find that as neurologists report, wheelchair-bound patients who are now walking again. It's not perfect, it's not good enough, but it's good. Okay, so, and that was approved by FDA in 2023, it approved in Europe in 2024. Okay, so, and indeed, 2023 was, in my view, a watershed year for ALS research, because there was evidence for slowing disease progression and partial disease reversal in 2 forms of inherited ALS, both from mutation in SOD1 and mutation in FUS. And if you just bear with me, this is for the scientists who bear with me for a moment, but there was one patient, Chris Snow, who was a hockey executive, who got press, got a lot of press. He knew he had multiple family members who had already died from this disease. He has the SOD1 mutation that's most frequent in the American population, and most patients do not live more than 1 year after disease onset. In fact, almost no patient. Okay, I've asked neurologists all around the world to find me a patient that lived more than 1 year with this disease after onset. I have yet to find one. So it's a very rapidly progressive disease. And here it is. So it's on CBS.
ALS treatment so far is something called tofersen from Biogen. It appears to slow the progression of a very rare form of ALS.
I'm coach sports. I'm play sports. I work every day.
Chris Snow got access to tofersen through a clinical trial. It's given via a spinal injection every 4 weeks.
I don't necessarily feel like myself or look like myself, and I act like myself.
Now, it only targets a particularly aggressive form of ALS, but it's just that also took the lives of Chris's father, 2 uncles, and his 28-year-old cousin. At the time he started on the new drug, he was given less than a year to live. That was almost 2 years ago.
He should be dead. That's the simplest way to put it. He looks pretty good for a dead person.
She says that too.
His wife, Kelsey, watched Chris get back on the ice and play hockey. He's the assistant general manager for the Calgary Flames. And he's still able to keep up with their 2 young children.
The quality of life that this has given us is really a miracle.
Tofersen hasn't completely halted ALS. Chris has still seen changes in his facial muscles. He can't really smile anymore, and he has trouble speaking and trouble swallowing. But many of his other symptoms have indeed slowed.
I don't think I'm dying. My fear is, can I play hockey with my son next winter? Instead, will my son have a dad next winter?
That's him teeing off for a round of golf just last month. And, yep, he played all 18 holes.
Okay, so Chris Snow, he died 4 years after disease onset from cardiac arrest, and the family released no further information about the details of his death. And the New York Times wrote a long story. Oh, I'm kind of thinking for the young folks, there used to be things called newspapers. And it would have been at least a half a page article about it, and they said that he got into the trial that was approved without evidence of efficacy. He lived 4 years after onset. It was spectacular evidence of efficacy for that mutation. Then there was an additional example from a second inherited form of ALS in the RNA binding protein FUS.
Athletic. She loved skiing. She loved playing tennis. She got weaker and weaker. She couldn't hold up her head anymore. She couldn't walk long distances anymore. She couldn't swallow anymore. Only at the moment that these genetic findings were explained to us, that it was a shock that it was ALS. The whole world stood still. In Germany, all the ALS doctors, they just said, no way of curing this disease. No hope.
You look at her and, she has extraordinary head control. She stood from a seated position on the floor with minimal help. She walks upstairs with, she walks around with her ventilator. I mean, it's really unmistakable improvement, and that's really lovely to see.
It was a miracle for us as well. The big hope we have now is that it works, that she can have a happy life.
Yeah, so Neil Snyder at Columbia initiated this with an ASO that was designed by Ionis, and so 2023 we had evidence ALS is a treatable disease if you have the right approach. And I just came back from the Oligonucleotide Therapy Society meeting in Budapest, and there I'm the keynote speaker today. I was the keynote speaker of that meeting in 2014, and the time that they had about 300 participants. The meeting in Budapest, there were 1,000. There were 100 new companies. Actually, if I can ask someone to get me a glass of water, thank you. The drugs that we put forward today. These are the Model Ts of ASOs in the nervous system. Thank you. It was very heartwarming. I thought it was heartwarming to see all of the bright young folks making modified oligonucleotides where efficacy periods were longer, and delivery being improved, including for the scientists, the strategy from Ractogen, a Chinese company, where to get their siRNAs into cells. This is not so simple. And the way they're doing it is kind of completely crazy. They're using the ASO uptake pathway. They're gluing an ASO onto an siRNA, and as crazy as that sounds, it really seems to be working. And so I'm absolutely confident that there will be 1, 2, 3, there'll be many new discoveries of ways in which to develop, to deliver these ASO drugs, these oligonucleotide drugs going forward. We know the disease can be treatable. Now, I did mention as well, though, that at the beginning, that if you inherit 3 copies of one gene encoding a protein called alpha-synuclein, you'll get Parkinson's disease. oh, there's an inherited form of Parkinson's disease. The most common inherited form of Parkinson's disease is in a gene called LRRK2. And the LRRK2 mutations, the LRRK2 encodes a kinase. It's an enzyme. And most mutations kind of depress an enzyme because it's a deleterious mutation. But these mutations activate the kinase. So a drug or an ASO that could suppress the gene would be directly on disease mechanism. And I was very fortunate, to get, in the introduction, you mentioned that I won the Breakthrough Prize in Life Science, and indeed, I was handed the prize by Sergey Brin, the founder of Google. And it was a huge missed moment of opportunity because it was publicly known, but I did not know that he carries the LRRK2 mutation. And so he's at risk for disease, and we went, Ionis Biogen went to trial to suppress LRRK2 in 2019. And that trial was completed by Biogen in December 2024. And disappointingly, Biogen has not released any information about success or failure. I can't tell you because they haven't told me. We just can't wait. And additionally, I mentioned that if you have 3 copies of the normal gene for alpha-synuclein, you will develop early onset Parkinson's disease. And so turning down that gene would be on disease mechanism. And by the way, almost all Parkinson's patients accumulate deposits in their affected neurons of exactly that protein. So this would be a broader therapy for Parkinson's disease. That too went to trial in 2020. And that trial was completed by Ionis Biogen at the end of December last year. And Biogen has not released any information about whether we were effective or not effective. Okay. And now, actually, Evangelos also mentioned this discovery of 47 years ago of a protein, the first microtubule-associated protein, tau. And we did show that it had multiple isoforms and that it had all the properties of a natively unfolded protein. We just didn't coin that phrase. But importantly here, that protein became much, much more famous than we had any right to imagine, because while there are 2 abnormal proteins or pieces of a protein that accumulate in Alzheimer's disease, one of them accumulates outside the affected neurons, but the tau tangles, deposits of tau are inside the affected neurons, and almost everybody in the Alzheimer's world agrees they are the best predictor of cognitive decline. If you had a room full of people with the extracellular deposits, A beta, if you're a room full of people my age, you'd find multiple ones of them who have A beta deposits, but that score cognitively normal, but you won't find anybody who's got tau deposits who's cognitively normal. And indeed, that went to trial to target tau in the initial phase of Alzheimer's disease called mild cognitive impairment. And that was reported, that you can see, in 2023. And to no surprise, you could lower levels of tau spilled into cerebral spinal fluid in a dose-dependent manner, or phospho-tau if you think that's the bad tau. You can lower it using one of these designer DNA drugs. But no real surprise on that. But the real question is about imaging. So there is positron emission tomography, PET imaging, to be able to find at least some of the tau deposits. And here are the 2 individuals that were entered into the trial. They were diagnosed with mild cognitive impairment, and you can see at trial entry right here, they have deposits. Both patients have deposits, and then they were imaged 6 months later. They were in the placebo arm of the trial, and 6 months later, looking at them, oh, the deposits got bigger. These people were in active disease, at least in terms of accumulating these tau deposits. And then COVID disrupted some of the imaging, the ability to image, but these two individuals were converted to drug phase and imaged a year later. And when they were imaged a year later, look what happened to the deposits. They almost disappeared. So if indeed that was a question about how long-lived would those deposits be, and the answer is, well, after a year you can get rid of them if you simply lower the synthesis to a third of the initial level of the target protein tau. And that was true in okay, it's a very small number, but that was true in 11 of 12 patients that were imaged. So that trial was run by it's an Ionis designer DNA drug. The trial is run by Biogen, and Biogen has not published, Biogen has not published the outcome on cognition. They did display one poster in one meeting in a very complicated way, much more complicated than necessary, but there was a cognitive improvement. But I'm waiting for the final publication so that everybody can see really how well or not so well that they did. Okay, so some of you were thinking, but he forgot, this is an ALS meeting. No, I didn't forget. So now it was my team, along with Kevin Egan's team then at Harvard, who discovered that actually one protein that is suppressed in almost all ALS patients is a protein called Stathmin-2. And so this was published in early 2019. We've subsequently demonstrated that Stathmin is an essential component inside neurons to be able to maintain their ability to stay attached to the muscles that they're going to trigger to contract. And it's lost in almost all ALS patients. And we know why. Mechanistically, we know why, because if you do a typical RNA-seq experiment using laser recovered surviving motor neurons from people who died from non-neurologic disease in the top, you see, for the scientists, you see exon 5, exon 4, exon 3, exon 2, apologize. These are the protein coding exons with the 3' bias, typical in these RNA-seq experiments. But in the ALS patients, recovering the surviving motor neurons, man, they are misspliced to truncate the RNA and mispolyadenylated within what should have been exon 1 of the intron 1 of the gene, making an RNA that really cannot encode a functional protein. And so this protein is lost almost quantitatively from almost all ALS patients. And we know, we've determined mechanistically how that occurs. We know that there's a TDP43, this RNA binding protein, binding to a site in intron 1, and it blocks the recognition of a cryptic splice site by the typical splicing machinery. But that then raised the question to us, well, we know that many RNAs are misspliced when TDP43, this RNA binding protein, which is now prototypically lost. You saw several of those slides this morning. Prototypically lost in almost all ALS patients. And is the loss of Stathmin, is that loss alone, is it something important? And so we tested that by taking completely normal mice, aging mice, these are a year old, and we started suppressing the ability to make just Stathmin and ask what happens. And if you suppress it for 8 weeks, you get about half of the neuromuscular junctions, the junction between the motor neuron and the muscle. They become abnormal. And it gets to be 2/3 of them by 8 months. You get the early steps of ALS from simply losing Stathmin. And so we argue that this is an important product, and that therapeutically, if you could restore Staffman synthesis, that maybe you could really do something good. And we showed, I don't think I put it, yeah, I didn't put it in. So, and if you take human neurons cultured in vitro, motor neurons cultured in a dish, and they have these long axonal processes, and if you damage them, if you cut them off, if they can make Staffman, they recover, and if they don't have Staffman, they cannot. So we argue that this is really a target, a drug target, and what we're going to do is to borrow a strategy that came from Adrian Krainer and Frank Bennett, the chief scientific officer of Ionis, where they showed in a childhood motor neuron disease that they could restore the synthesis of a missing product. The disease is called spinal muscular atrophy, but it's completely a disease of motor neurons. They just don't connect up to the muscles, and these kids, it's about 1 in 5,000 kids in this country born with 2 inactivated copies of a gene, and they just can't make that protein, and they're going to die typically by age 1. And Bennett and Krainer discovered how to restore the synthesis using one of these designer DNA drugs, a splice redirecting drug, and that became the first use of an antisense oligonucleotide, a designer DNA drug for approved for a disease of the nervous system, and the disease of motor neurons, and so. And Bennett and Krainer won the Breakthrough Prize in 2019 for that discovery, and we're going to borrow that. We were proposing that we can restore Staffman in sporadic ALS patients, and we can do so. We've made antisense oligonucleotides tiled across the region of where the cryptic splicing takes place, and we're going to start with a cell line which has a partial TDP43 loss of function from modifying both endogenous alleles with an ALS-causing mutation in TDP43. And sure enough, with the blue oligonucleotide, we can suppress the cryptic splice. With the red oligonucleotide, we can suppress the cryptic splice. And much more importantly, with both of them, we can restore the normal synthesis of Staffman and we've demonstrated that in cell culture. We've demonstrated by humanizing the Staffman allele in mice. We know we can restore the synthesis, and so we propose from that an on-target therapeutic approach would be to restore Staffman synthesis using a splice redirecting antisense oligonucleotide, and that strategy went to trial with a young startup company, Curalis, in 2023, and I'm hoping that I can get my corporate partner, Biogen, to get started this year. There's been some disruption at Biogen. They fired their CMO, their chief medical officer 2 years ago. They fired the CEO, and earlier this year they fired half of their scientists. It's a little... Anyway, we're still hopeful. I'm hopeful. Despite the data points, I remain an optimist. All right. So here's the scorecard for these designer DNA drugs in nervous system disease. It's an approved drug for spinal muscular atrophy. Those kids that typically die by age 1, the oldest ones are now 7 or 8 and walking. It's really, for the neurologists, the child neurologists, they know, you see these physicians who are really seem tough. They kind of tear up a bit when they realize that what used to be completely hopeless now really does have hope. I showed you the evidence for ALS from the SOD1 mutation, I proposed, and there's a trial now being initiated by Ionis to suppress FUS, building on the work of Neil Snyder. And I proposed for sporadic ALS, restoring Staffman 2. Oh, additionally, there were 2 other products. The largest genetic cause of ALS, as Dr. Isaacs mentioned this morning is a nucleotide expansion within the gene C9orf72, and that trial failed. 2 trials failed, one from Ionis Biogen and one from Wave. And there's I mentioned from the microphone that there's an intra-team disagreement on the source of that failure. I think the truth is we do not know. And I have been negotiating with Biogen to try to get the remaining samples, the autopsy samples, to try to use an approach I'm about to show you, which I think we can maybe parse out at least maybe something more about why that trial failed. Additionally, there was a second one for a target that was put forward by Aaron Gittler at Stanford, a taxon 2, and that trial failed, but I think that was really a true trial failure. They really did not suppress a taxon, therefore it was a drug failure, not a hypothesis failure. Okay, so what's next? And I'd like to tell you 2 futuristic stories. One which it's football season, and one which I'm doing this a little bit in jest, but actually I'm quite serious about it. And then additionally, one of the grand goals of neuroscience for 100 years is can we make new neurons in the aging nervous system? Okay, so, but first, everybody here will know about chronic traumatic brain injury, CTE, National Football League disease, and that the founding for the oldest timers, the founding case of Mike Webster, and then in San Diego, the most famous one with Junior Seau. And we now know that there are hundreds of these individuals who have died from CTE, and it's diagnosed post-mortem by examining the brains. And what you see are these focal deposits, the brown stuff. A focal deposit, here's another one, here's another one, here's another one. And this is the work of Anne McKee and her colleagues at Boston University. She had season tickets to the National Football League. She was the go-to neuropathologist that the NFL did go to. And, that was before she diagnosed, in the first 100 football players that were looked at, 99 out of the 100 had CTE. Now she's persona non grata at the NFL. But it's these deposits, this brown stuff, and if you look at a close-up of one of those, it looks like a little starburst stuff, this brown stuff, like it was a focal injury that was kind of spreading. Oh, and the brown stuff is tau. It's uniquely, it misaccumulates in Alzheimer's disease. It is the protein that is known in chronic traumatic brain injury. And the safety trial has just already been completed in suppressing tau in mild cognitive impairment. So my fantasy is that we're going to be treating football players with their prophylactic doses to permit them to continue with their brutal game, and without the likelihood of CTE. Okay. Now it's now Tom Brady. He said his brain understands the position he's in, and so he's okay. And I'm thinking, well, really, maybe a non-target therapy would be to reduce tau synthesis in those individuals, and so I'm actually really serious that this could be a prophylactic approach for CTE. Okay, so now one last little vignette. And so it was the greatest neurobiologist of all time. It was Ramon y Cajal, Spaniard. And he noted back 100 years ago, more than 100 years ago, that you just don't see new neurons in the nervous system. And he challenged the next generations. He said it's their duty to find a way to overcome this intrinsic failure to regenerate neurons. And some of you And there are two known regions of the brain during development and early life, postnatal life, where neurons are born. There's the dentate gyrus of the hippocampus, and the major one that makes the most neurons is the subventricular zone right here. And indeed, this is called the ventricle. Okay, so that's where neurons in early life are made. And now, just for the scientists, just this July 3rd, there was a paper claiming that there is new neurons being made even in the aging adult hippocampus. And that was followed up by a newsy article from an editor of Science that said, this evidence may close a 100-year-old debate. Okay. I looked at supplemental table 1. There were 21 autopsies there were 21 brains that were examined, and the claim was that they continue to make neurons during aging life. And here they are. Here's all 21 data points, and most of them are 0. No new neurons. The second most frequent number is 1. And then there are 2 outliers that I put in blue. So I think a better interpretation of this would be that there is no neurogenesis in most aging people, but in some cases, maybe you can make new neurons. And indeed, the most frequent cause of death of these 21 people was suicide. And no doubt they were taking things, either conventional drugs or street drugs or something. So I think you could make new neurons, but actually most people don't make new neurons. Okay, so can we now use designer DNA drugs to generate new neurons in the aging mammalian brain? Okay, so the way we're going to test that is to use a spatial transcriptomics approach. Homemade, and you can see here's the front end looks like a microscope, but the back end has got a lot of fluidics. And this is built by my faculty colleague, junior faculty colleague, Bogdan Bentu at UCSD. And with it, we're going to be able to, we can determine the rates at which every gene in every cell is made, almost complete transcriptomes generated. And what we're going to do is to make, there are 50 different probes for every gene. We're going to do 2,000 genes, that's 100,000 probes. And you hybridize them to your tissue section, there they go. And now they're glued onto the 5' end of the oligonucleotide is an acridite linker, so that for the scientists, you can cast a really thin acrylamide gel, and you now anchor those probes in place. And you don't care that the RNA gets degraded in subsequent steps, because we're just going to read out those 100,000 probes in sequential steps. And if you do that, again, we only got 3 colors to read out 100,000 different probes. So it's a very complicated combinatorial readout, but you can do it. And here, to show an example of that, if you look for a gene that's expressed in inhibitory neurons in red, and you look, in excitatory neurons in red and inhibitory neurons in green, and you look the first round, you're counting up individual RNA molecules for that RNA. You can see these are 2 neurons adjacent to each other in a tissue section, and one's red and one's green, and even 74 cycles later, it's almost identical. We can do almost limitless cycles so the depth. And now for the scientists, you heard some about single nuclear or single cell RNA-seq this morning. The read depth of this RNA-seq is a little bit, by today's standards, a little limited. We know that a gene marker that everybody uses for human lower motor neurons is a gene called CHAT. It encodes a protein that's involved in making the neurotransmitter that lower motor neurons use to trigger muscles to contract. And there's about 600 RNAs in a single human motor neuron. And 10% of them in the nucleus. But there are two competing data sets. Some of you know this. There are two competing data sets. It's a he said, she said story about who's got the best of these single nuclear RNA-seq ones. And if you ask how many chat reads do they have per neuron, the answer is, in Aaron Gettler's data set, 1. Actually, the most frequent number in the Gettler data set is 0. And in the competing data set from Levine's team at NIH, it's 0.01. Those are not motor neurons. I don't know what they are, but they're not motor neurons. But you can see our read depth is between 50 and 500 times the read depth of the current RNA-seq data. And if you do that, and you take a slice of brain, and you say, can I identify the cell types in the brain? And again, the scientists will know these are the UMAPs, and you can see that there are 68 different cell types. But because we have spatial, we can look at the transcriptomes. We can name all the 68 cells. And we can see where they are. And in the founding image, there are 22 kinds of neurons. There are 8 kinds of astrocytes. Remember that search for astrocytes? Ben Barris had finally found A1 and A2. There are 8 of them right there. And there are 7 that are localized in different regions of the brain. And the 8th is everywhere. It's probably a precursor. Okay. And why do I mention that? Because I'm going to look for new neurons using this approach. And so here, we're going to look in the ventricle, one of the 2 neurogenic niches, and we're going to ask in a young animal, look, there are ependymal cells, the green cells, they're lining both walls of the ventricle, but lining one wall are these yellow cells. They look like neural stem cells. They have the transcriptome of a neural stem cell. Okay, and then you can see in a young animal, they're turning into neurons, immature 1, immature 2, the blue neurons and the purple neurons. Okay, so, and here, if you look at postnatal day 5 in mice, you can see, oh, there are a lot of neural stem cells, the yellow cells, and they, my God, here they're both in the dentate gyrus and they're in the ventricle here, and they're just streaming out everywhere. They're making plenty of blue cells, plenty of young blue and purple neurons. By the time you get to 4 weeks, they're still making them. You can see that there's the stems, the yellow neural stem cells. They're making immature blue neurons and immature purple neurons. But if you get to 1 year old, now there are a few purple neurons, but there are very few. Okay, so, but there are lots of yellow cells. There are neural stem cells that are sitting there dormant. Can we use an antisense oligonucleotide to wake them up? And so we did that using a proposal from one of my former colleagues was that that cell identity, neuronal cell identity, the first step is a 3-gene circuit where each gene inhibits the other genes. And so if you have the one that's present in the most, if it's an inhibitory battle, will win. Okay, so, but immature neurons, they have a lot of one of these, the orange one, and they have a little bit of this one. So maybe you can convince these things to turn into neurons from a dormant stem cell into a neuron by transiently suppressing the synthesis of it's called PTBP1, an RNA binding protein. Okay, so here you go. 8-week-old mouse, gosh, it's making lots of young neurons. Oh, and here's the ventricle. They're streaming out of the ventricle, going this direction toward the striatum. Okay. 1 year old, oh gosh, there are only a handful of these pink neurons. The yellow cells are still there, they're just not making neurons. And if you transiently suppress PTBP1, in both of the 2 neurogenic centers, look what happens. You make a lot of these purple immature neurons. They stream out from the ventricle, just as before, and they make them in the dentate gyrus as well. Okay, so we can produce new neurons by taking dormant precursor cells and transiently using an antisense oligonucleotide to suppress one gene, PTBP1. And now, lastly, yeah, so you can do that. Do they really become neurons? They stream out from the ventricle. They stream out toward the striatum. And actually, I'm summarizing a lot of the work here in this slide. They undergo a cell division first. They become, if you use a thymidine analog, EDU, you see that they go through DNA replication. At the end of making new neurons, there are just as many yellow cells as there were before. It's just that there's a bunch of them that became pink, blue and pink, and then streamed outward. And if you do that, and you say, well, if you look in the actual striatum itself, here's seven sections of a striatum, and you can see, oh, there's a few. We know that they're neurons because they have the transcriptome of a neuron. They're EDU positive, and they're kind of scattered around. And EDU positive, they're really EDU positive. This is not DNA repair. They're really EDU positive or not. And if you count them up and you say, well, so how many EDU positive neurons are there if you do or don't suppress PTBP1? Well, that's easy. There are 0 EDU positive neurons if you don't suppress PTBP1, and there's a whole bunch when you do. And if you look at the transcriptomes of those neurons 1 month after converting from a stem cell to a neuron, and you're at the level of 2,085 genes, and you can determine the nearly complete transcriptome there, and you look at the mature old striatal neurons and the new EDU positive ones, they're nearly identical. So they have achieved mature transcriptomes. I know you're going to say, yeah, have you shown that they're electrically active? No, we haven't shown that yet. But they are in the right place, and they've streamed out there, and they really have adopted, in a very short time frame, a nearly mature frame. And actually, the last part of that, well, what about the other neurogenic niche in the hippocampus? And the answer is, well, if you look for young neurons, markers of young neurons, there just aren't any in old, this is a 1.5-year-old mouse, there aren't any, but if you suppress PTBP1, look what happens. You make new neurons. And those new neurons send, we know that they had glial character to start with, as the stem cells do. That we can make 8% of new neurons in the dentate gyrus from a single dose antisense oligonucleotide injection. They are electrically active. If you look at the new neurons versus the previous neurons, their electrical properties are almost indistinguishable. And indeed, if you do these mouse cognition tests, I know they're really anyway. They learn a little bit better. Okay, so we argue from that we can make new neurons in a therapeutically viable manner today if you're a rodent. But you may care, what if you're a human? And so where we are today is that if you now take a postmortem sample of human brain, here's the ventricle, it's collapsed, postmortem issues with human samples, but you can see the yellow cells, stem cells, line the one wall of the ventricle just that you've seen before. So we argue from that it's plausible that actually the neurons are there, and now the next test will be in a non-human primate. Can you actually generate new neurons in a therapeutically viable way today by transiently suppressing PTBP1? And we propose, haven't demonstrated, that we can do it again and again and again. And so that this could be a way of generating new neurons to replace those lost to disease. And the work I described, here's the founding quintet for the use of an antisense oligonucleotide in the human nervous system. Richard Smith, who was adamant that we get started. Tim Miller, who's now co-run the ALS trials. My animal cell coordinator who did the surgical manipulations to introduce these antisense oligonucleotides into mice and rats. Frank Bennett, the chief scientific officer of Ionis. That we moved to Huntington's disease. That work was done by Holly Cortisewicz. And for ALS, for Stathman, this is the big team. Two former postdocs, 2 former graduate students, demonstrating that we can restore synthesis of Stathman in nervous systems in which TDP-43 has been suppressed. My long-term partner, Clotilde Lagier-Turian at MGH, and for FUS, Sandrine de Cruz, who there wouldn't have been a FUS ASO to go forward if it hadn't been Sandrine. It was she who got the successful ASO that Schneider used. She did that by just insisting that Ionis make it. Okay, and then lastly, here's the team for answering Cahal's challenge of making new neurons. A quartet of Spaniards, plus Roy Mamon, who's about to take a job at NYU. And my colleague, Bogdan Bentu, who has built these very, very capable spatial transcriptomics approaches. Thank you very much.
All right. Thank you, Don. Are we going to take questions now, some scientific questions, or are we going to move straight to Q&A after?
I think we should take a break because there will be a question and answer session already, if that's okay. But if there's like a burning question, maybe we can wait after the break, and let's give a round of applause to Dr. Cleveland again. Gift for him. Dr. Cleveland, I have an art gift for you. I took one of your photographs and I turned it into a personal art. I hope you like it. This is your photograph. You can see it, I hope.
Yes, indeed. It's
and
I think I have it upside down. I still have it upside down. I don't know.
It will always be right.
It's always right. Yes. Thank you.
It will always be right.
Thank you.
Thank you so much.
Yeah.
So she's going to take a photograph.
Oh, yes.
Okay, so there's a short break and then there's a Q&A session right after.
So we will be starting the afternoon session, and please be seated. And Dr. Sanda will have the microphone soon. And thank you so much for staying. And please write your questions on the chat for the online viewers. And Dr. Sanda Driss, the ball is in your court.
Well, welcome, everybody. You gave us a very nice overview of, I feel like, 3 decades of research there. Before we go into the actual Q&A, I was wondering if there is any question in the audience first, because we didn't have a chance for the audience to ask any question. Anybody? Yes.
And Tom, I think, when I saw the A4V mutation patient treated with tofersen and lived for up to 4 years, I think that is really amazing. So my question is, so that is the ASO. And for ASO, we have to, of course, from the therapeutic effect, we can see the therapeutic efficacy is over there. There's no problem. The issue is we have to repeatedly to inject.
Yeah.
So how often did the patient receive the injection of the toxin. So that's one. The second question is, do you think the microRNA and or a siRNA, because those can be delivered through AAV, and it seems they can live for the muscular atrophy, spinal muscular atrophy for several years. So whether that one actually will be better than the ASO from the administration point of view, or combine both of them.
Yeah, so there are actually multiple questions in there. So for today, there is no it's tofersen or not. Is tofersen a great drug? Is it going to be the long-term drug? Oh, absolutely not. It's going to be overtaken by better oligonucleotide approaches, including, I mentioned one, this company Ractogen that has glued an ASO onto an siRNA and is showing 2 or 3 times the length of efficacy period, and claiming that that it's really non-toxic. So, and then I was at the Oligonucleotide Therapy Society meeting. There were multiple companies with modified siRNAs arguing for 6-month, 9-month efficacy periods. The problem for them is the promise is great. They haven't really demonstrated that they can dose them broadly in the nervous system. And that gets to the second part of your question, which is, well, why don't we just use an AAV to encode an siRNA, and then you could do, as in SMA, there are 2 approved drugs. Both have efficacy. One has to be redosed, the ASO, and one is a AAV for which there's it's one dose and done. Actually, my reading of the current literature, there's a disagreement about how broadly in a large animal can you really get transduction with AAV, and what's the ideal method of delivery that's going to target at all of the or many of the cells that you really want to hit. And if you look at the published reports, you're going to see that I'm right. That's because my name is on is on 2 papers claiming diametrically opposed things about how effective the ASOs can be when introduced intrathecally and in a large animal. And I'm completely confused by 2 very capable teams, one of which says you can transduce broadly, and the other says you can't transduce broadly at all. So having said that, there are many young AAV companies making alternative capsids, and I'm absolutely confident that one of them or more of them will produce an AAV that will be much more effective in terms of delivery, in terms of targeting the cells that you want. That it's going to happen. It's a process, though, that takes time. And I think it takes time and open-mindedness, because some crazy idea may prove to be the best way, and that we should be we should really need to be open-minded to alternative approaches. I think I said, and I really mean it, that a drug like tofersen, this is a start. It's not. It's not the final. There'll be many improvements, but as the audience knows, it takes time to demonstrate something that really is effective. So there will be 5 years before there's an additional SOD1 drug, in my view. Novartis abandoned the siRNA AAV approach. I know they abandoned it because they had licensed the patent. It's Brian Casper's patent but I'm on the patent. And I got notified that Novartis was abandoning it. So, and Brian Casper bought it back. So there will be an siRNA to SOD1 going forward. It's from Insmed. It's some point in the future. I don't know the timing. And there definitely will be Ractogen is in an early stage trial in China to take an siRNA to SOD1, but delivering it by linking it to an ASO, so that the ASO uptake pathway provides for better siRNA entry.
So that's my second question. That's the third question. How did they, pack the microRNA, the siRNA with the ASO? I thought the ASO normally we are injecting like intrinsically, and microRNA normally we use the AAV to deliver.
I'm just saying. I'm just reporting the company Ractogen is founded by a guy Lee. He used to be on the faculty at UCSF, and he's now the CEO of this company in Suzhou, China. I just saw him. The data are really quite good. Their homepage describes the use of an ASO to be able to more effectively deliver an siRNA. It's to me, it's completely counterintuitive, but the data look pretty good. So I'm open-minded. Would be, young folks are gonna make the discoveries.
That's right. Right. Thank you.
And there are going to be plenty of them.
So we're gonna, another question, okay.
I have a question. Hi, thank
Hi.
you. I know for a lot of SOD1A4V families, there's a huge difference in age of onset.
Yep.
I'm also here to tell you, everyone should read this book. It's called The FAR Disease. It's about a family with an A4V SOD1 mutation who has a history of paralysis in their family going back to the 1700s in Vermont. And they present a really great example in this book of a mom who survives until her 80s. She's an A4V carrier, has no onset, and a son who then inherits that gene from the mother and dies by 26. And so I guess that's my preface to this question of, if you are a member of an ALS family, you want to get into a preventative trial with tofersen, which I know has been like a topic of conversation. At what is there risk to doing that too early, right?
Ooh, it's a very, very thoughtful overall. Thank you for telling about the families, and absolutely the A4V is the most prominent mutation in North America, and I thought you were going to ask me how come we don't know what determines age of disease onset, but it was implicit in your comments, and I think the answer is we do not have a clue as to why that is. And I'm certain that I do not have a clue as to what that is, but I think that's really true for to the field. Now, in terms of the trial that is ongoing to I forgot its name, to dose
Atlas. The Atlas trial.
Yeah, Atlas trial. Okay, so there you go. So what are the criteria for getting into the Atlas trial?
So you have to be a carrier of a fast progressing SOD1 mutation, but there is a lead-in natural history period to it where they follow neurofilament, and then only when the neurofilament reaches a certain threshold that they're enrolled in the trial. Yeah,
so we have one with actual clinical experience and just an outsider on that. I would have thought that's pretty sensible, that if you see that the levels of neuronal damage, NFL, you see it going up, I would have thought that's a pretty good time to get started. And that now the trial will test is, can you really slow slow disease advance? And
Yes.
I'm thinking yes, but
We'll find out.
I think it makes really good sense. And the same would have been true in, we were thinking about that in Huntington's disease, except the trial completely failed, suppressed the mutant gene with no clinical benefit. Really, really, really surprising, disappointing. Yeah.
So just to move on a little bit, what I wanted to do is have a very candid conversation with you and really put it in words. Oh, there is another question. Okay.
The Professor Clement presentation. So about the new neuron generation that you were talking about in the presentation. So how far they were resistant to the pathology that is currently going on with the, for example, ALS patients. So how far those new neurons are resistant?
So actually, I should have clarified. I do not think that replacing motor neurons is the right place, is the place to start. In fact, those, meter-long neurons, no, we're not going to replace those. It's going to take forever for those. Even if they knew where to go, they can't go the right place. But in Parkinson's, there's been many efforts to try to inject cells to hope to get replacement nigral neurons, and I have not demonstrated that we can make those. Although I really want to be able to do that. We can make striatal neurons, and now so for Huntington's disease, maybe that's the starting point. Mm-hmm. It's actually I usually say when I talk about this, I say, well, I can answer the question before you ask it, because I know the answer. The answer is I don't know, or we don't know, and it's really early days. But what I'm confident of is that we do have dormant precursor cells that would score by their transcriptomes as neural stem cells. They're just dormant. And certainly in mice they're dormant, and certainly in of one human example. Oh no, there are no new neurons in that example. They're yellow cells. There are no blue cells by transcriptome. There are no pink cells by transcriptome. There are none. It's just, but there are yellow cells. There are a lot of yellow cells, and so I'm inferring from an N of one that there really are dormant cells present, and that we maybe could wake them up. And so non-human primate experiment has not been attempted yet, but actually that's not quite correct. It has been attempted. It was attempted in a Parkinsonian context to pair a normal side with an MPTP, a damaged side, and now determine whether you could make new neurons by suppressing PTBP1, either on the damaged side or on the control side. And those primates have been collected. Not a single merfish image has been produced today. And I mentioned, I think privately, the merfish has, at least the platform that we're using, I do think it's enormously powerful. It's enormously slow. Really, so throughput is really a problem. But right questions, we just don't have the answers.
Thank you.
Thank you. Okay. So my patient always asks me, why is it taking so long? I mean, it's been 30 years since we discovered SOD1, and it took about 3 decades to find an answer.
Because it takes a long time, because even when you have you know what the right target is, and even when every part of the process, it must be proven at human trial. a good idea is good, but it's not good enough. You have to go to trial. The trial design takes time to implement the patient recruitment, and then to read out. So, from trial initiation to completion, it's 2 to 3 years. It took you. It's appropriate that for the regulatory agencies that you have to demonstrate the safety initially, that you need preclinical toxicology. Yeah, I know it's frustrating that it's so slow. We started this in 2003, and we have success in 2023. Yeah. So it took us 20 years, and we thought that we were going, and it was kind of implicit on one of my slides. We discovered Stathman as a target in 2019, 2 teams did, and we showed proof of principle that we could restore Stathman synthesis in a TDP-43 independent manner using an ASO in 2023.
Faster.
And so we thought 4 years from target gene identification to trial entry, and this small company, Curalis, did just that. They went to trial, and I'm partnered with Ionis, and Ionis had already partnered with Biogen, and Biogen has the rights to the Ionis product, and Biogen's in a mess, and Biogen has not made any decision on what to do, and sometimes things take a long time because your partners get disrupted for some reason or another. Yeah, and it's super, super frustrating.
To follow up on the statement, do you think with all the other misplaced kind of product, just correcting one would make a big difference?
So I do think so, and I think there are multiple examples where I think there's, and Adrian Isaacs mentioned it this morning, but I think there's almost universal agreement that the gene that is most sensitive to TDP-43 partial loss of function is Stathman. It's true in almost everybody's hands. Adrian has an opportunity to give a contrasting point of view, but I think it's true. And we of the misspliced products that have been identified so far, the Stathman, we know that you will get denervation by just losing Stathman. So, of course, restoring full TDP-43 function would be an optimal way to go. Yeah. But I would also counsel that we shouldn't let the perfect be the enemy of the good, right. And that the tofersen is good. It's not good enough. It's going to be superseded by better drugs that can suppress mutant SOD1, and those will be superseded by even better ones. I got it. But so we're we start with since we showed that we really can restore the synthesis of Stathman. Curalis went forward without evidence in the mammalian nervous system that their drug can restore Stathman expression. My clinical partners, corporate partners, would never have gone forward without that information. I hope that they're right, but there are plenty of ASOs that work in cell culture, that work on peripheral genes when introduced in the periphery, that are not effective in the nervous system. And, no, we don't understand that.
Right.
And since I know it continues to occur in candidate drugs that Ionis produces, whatever the algorithms for identifying effective ones or ineffective ones, they're not good enough.
Yes.
To eliminate those disappointments. So it takes time, and you need real evidence. The two failures of ASOs in a clinical trial, unhappily, are in the larger the diseases, Huntington's and C9orf72, and they're in repeat diseases. And I think a fair view of them is that we're confused. The big W on we're confused as to why they failed so miserably. And even for C9orf, for this audience, for C9orf72, as Adrian described this morning, it's bidirectionally transcribed and possibly targeting the sense strand it certainly left the antisense strand untargeted. Maybe the antisense strand is actually the bad one, and that maybe even if you have co-transcriptional, if you're really making both strands, don't you have machines running, RNA transcription machines running both directions, and it's possible that the catalytic RNA destruction that you get with RNase H, maybe you actually even disassemble that machine, the forward machine, which means you have no impediment to the antisense machines. Those things have to run into each other, and so maybe we actually did the wrong thing. Maybe antisense strand is the bad stuff or the worst stuff, and what we did was to turn it up by turning off the sense strand.
Yeah.
That none of those is the current data set does not allow, you can't distinguish among many aspects. And I meant to say this morning, but I failed to, that I've been negotiating with Biogen to try to get the samples that they have from the C9 trial to now be able to look using the spatial transcriptomics approach to really look to see, did we really suppress things, and which cells did we suppress things, and in the same cells did antisense things go up, did the DPRs go up, and the technology that I described where we called it multimodal MERFISH, we can get the nearly complete transcriptomes plus of all cells in a tissue section, plus then use immunocytochemistry to look for up to 10 different proteins. Oh, and we can measure repeat length. So I ask about somatic expansion. In principle, we can measure somatic expansion. We absolutely can do so in Huntington's disease, where it's a trinucleotide repeat with an a CAG. For the GC things, in principle, but not demonstrated, we can measure somatic expansion. I hope we can. We have not attempted that yet.
Another question that my patient asks is, how important is it really to study familial ALS, because they're only 10% and 90% is sporadic. I think during you answered that. I want to ask you a question that somebody asked me on my way to the restroom earlier. Misfolded SOD1 could be seen in sporadic ALS, so why not choose the first in sporadic ALS?
It's controversial whether there is misfolded SOD1 in sporadic ALS. I'm on the side that there is not. I removed my name from one of the papers which made the claim that there was misfolded SOD1 in ALS. I thought the data much more strongly supported the absence of misfolded SOD1, and so I'm not I just don't. Now, I think the rationale based on today's evidence is quite weak for treating sporadic ALS patients with something like tofersen. On the other hand, there's not, we don't have effective drugs, and there is a controversy about how you interpret immunohistochemistry, and I personally think it's one of the world's worst quantitative tools. And yet some are trying to use it as a quantitative tool.
There's one quick question here.
They say is Curalis trial the only one doing human trials targeting statin 2 right now? Is this the only trial?
It's the only one I'm familiar with, yes.
Okay, thank you.
Yes.
So your statin 2.
Yeah, there's an online audience, Sammy, so.
So in your statin 2 trials, you showed that there was a increase in, when you did an RNA-seq type experiment, elevation of a number of genes. Interesting, because they all turns out to be in microglia. And so the question I have is, when you have these new cells born in the ventricular zone or in the hippocampus, they got no targets. Like those targets are developmentally determined. So my argument is going to be is that these cells are going to die, and that's why when you look at your RNA-seq data set, I just very briefly looked at your volcano plots, and the first thing that came up to me were a bunch of microglial genes. That's because they want to clear the stuff that's not going to make targets. So if you want to address that question, I mean, is it really relevant? Is it worthwhile doing this experiment to continue like making new neurons in the adult brain, which have no real targets?
So the question is, if you make electrically active neurons that have mature transcriptomes, will they be functional? That's Sam. This is Sam Sisodia from University of Chicago, the other university in Chicago.
The only University of Chicago.
Yes, it's. So of course we have.
And I was one of his first postdocs, just so everybody knows.
Worse yet, he's
before the neuro world.
He's digging a huge hole. It was there was a dinner last night, and I said that one of my the way we got into the ALS altogether was because the SOD1 mutations had just been published, and we had just demonstrated that if you make mice with too many of the structural protein neurofilaments, that they die from fatal motor neuron disease. And that was published almost contemporaneously with the discovery of these SOD1 mutants. And one of my cocky postdocs said, we should work on that. That was Sam Sisodia.
I'll take credit for that.
Yeah, so it's the earliest days of trying to of determining, what can these things really become. In the Parkinson's world, they've been injecting cells now for 30 years with the hope that they'll turn into dopamine-expressing replacement neurons.
Yep.
The literature is, let's go with mixed on whether that any of those have been successful. In the Parkinson's world, there are many now, Studer in New York, they're going forward with new trials, and I would have argued, why don't you just, if that can work, why don't you just make your own? And instead of using surgically implanted cells, which is a big deal, we have not demonstrated that these new ones made, the ones that have nearly mature striatal transcriptomes, do they have any real function? You propose that they were on their way to death. I'm not buying that, but
we'll figure it out.
Dr. Sisodia has been right occasionally.
As have you, Don.
So if there is no other burning question, I think we are at time.
I have one question.
Yes, please.
Actually,
Use,
it's a good way to...
use the microphone.
Go to the microphone.
It's actually not really a scientific question, but to end this symposium, I think I would just love to hear from you as somebody whose husband has been living with ALS for the last 11 years and also speaking to so many newly diagnosed people. what should we be hoping for, or what should we be hopeful about right now in the field of ALS in scientific research and discoveries?
So the scientific discovery worldwide will make new discoveries. They'll always be too slow for patients, their families. I know that, but we're going to. Their progress is going to be made. Progress in the U.S. has there's a little hiccup from the current administration, which is, it's more than a little hiccup. It's a huge hiccup, and we're not sure how badly it will be disrupted and for how long. It's a very destabilizing effect. I do think, actually, on a very upbeat note, I do think that the Curalis trial has a real chance to be a therapy that can be of high benefit to sporadic ALS. They also have a significant chance of total failure. So that's why you go to trial. And they went to trial, maybe they didn't know that ASOs, from the way we test them to the actual mature nervous system, some of them don't work at all. And they may not have known that, or they may have just thought, we're taking the risk, because there was no appropriate animal in which they could be tested. We built one where we humanized the allele, or the gene, so we can now test whether they really work. And but I think they have a chance. They have a real chance. And we'll see. I thought we had a real chance on the most frequent inherited form of ALS from this expansion in C9orf72. The trial failed, and we're just really confused why it failed. It should have been good. It shouldn't have been no benefit at all. And that's we as a community work really very hard. And yet sometimes we have failures. Sometimes we have successes. So for the approach I described, so far there have been 4 clinical outcomes. We have 2 successes, 2 failures, which in the pharma world, actually, it's like in baseball. If you're batting 500, you're making it, or you're batting 400, you're right. You make outs more often than you don't, but that's actually a super impressive number in pharma. So I think there's a reason for some optimism, and for the sporadic ALS patients, I think the Stathman trial could be valuable. Won't be perfect, but there are those. The New York Times has been one of them, who has relentlessly said that the SOD1 drug was approved without evidence of efficacy. Post-approval, there's tons of evidence of efficacy. So stop saying that it's not effective. It is effective. It's not good enough. And I am predicting. I saw the data twice in the last 2 weeks. I saw the updated data from this company, Ractogen. They are going to have they're going to have a product that will be more effective than tofersen, that appears to be as safe. It's in the early trial. Whether it'll be, because it's a Chinese drug, will it be approved broadly in this country and in Europe, and how quickly? Oh, that's a different issue, and one about which I have no expertise. But I know that there will be better drugs, and there will be steady progress. I'm hoping that if the Curalis trial actually shows a clinical benefit, oh, that would be fantastic, just fantastic. It could. There will be other targets, and there are multiple other approaches toward trying to, in sporadic ALS, the loss of this protein, TDP43, loss of function
It's broadly true. I think almost everybody agrees that that's the case, and it's a major component of disease. May not be the only component, but it's a major component. There are multiple strategies to restore it.
Yeah.
Somebody will succeed. I've got a strategy. I've got two of them to restore it, and we'll see. Can we make who is going to be the first to make one work, and the second one work? It will happen, but it will be a decade. I'm sorry, it just takes a decade from today. It'll take us the strategy that I'm familiar with. It's going to take 2 years to get them to the point that pharma will really start, and then there's a 3-year process of producing that. And then there's 2 to 3 years of trial. That's a decade.
What can patients do, and families and advocacy, to move this faster?
Well, it's certain. I really do think that, for example, with the discovery of Stathman, which is lost in sporadic ALS, and that going to trial within 4 years of a discovery was about that. Actually, I don't think that can be accelerated much. Today, trying to ensure that American science and biomedical science continues to receive bipartisan support across the country, as it has for 50 years. Mm-hmm. That, I think, could be something that everybody could participate in. If you live in a blue district, if you live in a red district, mm-hmm. let your representatives know that support for biomedical research is part of citizenship. It has been for 50 years. America has been the unrivaled world leader in developing new medicines, and we should not abandon that. And there's just no reason to. No matter what your politics, there's just no reason that we should be stepping back from that. We can differ on lots of things, but the world leadership of the United States in biomedical science we've been unrivaled, and we should not abdicate. And
well, thank you. Yeah, go ahead.
No, and our representatives, call them, meet them. There will be many on the Democratic side that will just need reinforcement that their supporters will continue to support them, and on the Republican side, the leadership. There's a core of Republican congresspeople and Republican senators mm-hmm. who have been very strong supporters of the biomedical research in this country, and they need help to be able to feel that they can survive in the current climate and still support science. And before the government closed, there were competing bills passed by the House, passed by the Senate, and they were in reconciliation. And they were the Senate bill was far more favorable to continuing biomedical research, and again, maybe even more so if you in a state in states that have Republican senators, getting a few more votes could really change the course of biomedical research, and enthusiasm for continuing that, I think, could really, I think it could really make a difference to maintaining the biomedical machine and extending the discoveries that are made. Yeah.
Thank you very much.
There's one last question about biomarkers.
So, Don, I applaud you for incredible advances in.
Who gave him the microphone?
Hold on, just a second. But my question really relates to so treatment of people with ALS, familial or otherwise, and the C9orf failure really struck me, and I wonder whether you begin these treatments too late. So so tell me what's going on with biomarkers. Wouldn't it be useful? Like, I've not heard anything about biomarkers in ALS, and it would really be interesting to know what kind of biomarkers, blood, brain, CSF, biomarkers you could use as a sort of a surrogate for like where you are in progression of disease, that you can treat the right people, so you might actually get an effect. So why C9orf being the major gene for early onset and certainly for late onset disease? These people are not they're not responding. I don't know, has anybody actually looked at the pathology of these people who died after the treatments?
Yeah, so yes and no. And so there was, Jonathan Glass published a paper in Cell last month, and Adrian mentioned it this morning. I have a less favorable view of it than Adrian presented. I really, I think we as a community really don't know why it failed. I think the highest likelihood myself, there are multiple points of view, and Adrian talked about maybe really the bad thing is the antisense strand. We targeted sense strand, and that certainly is a viable hypothesis. I think the most likely one is that the drug taken forward had toxicity, and that they covered up any potential efficacy by dosing it at a level that brought its own toxicity. And there are multiple ASOs that when introduced that are beautifully safe when introduced in the periphery, that are I mean, not just a little bit, but wildly toxic when introduced into CSF. And we don't fully understand that. They, of course, are screened for that. My personal view is that the trial failed because Ionis made a toxic drug. And maybe to your question, it's being tested. It's being directly tested by Roche, not in ALS, but in Huntington's disease. So there were at least 3 ASOs that went to trial to suppress mutant Huntington, one from Ionis Roche, and 2 from Wave. Wave has a trial that's continuing now, but the first 2 Wave ones, there was unambiguous. They know how to make toxic drugs. And now, but the Ionis Roche one, again, the possibility is that they were dosed at a level where there was a toxicity from the drug that covered up any clinical benefit. Roche is testing that. The first trial was a 700-patient phase 3 trial. There's a second phase 3 that's being run with exactly the same drug, where they're going to dose less, and they're going to insist on patients being much earlier in disease because the fear that maybe you started too late. Yeah, so at least for one repeat disease, there's an ongoing phase 3 test, 350 patient trial by Roche. So it's easy to be critical of big pharma but at least Roche is really they're putting their money. So we'll see. These drugs, tofersen, the drugs are a little close to maximum tolerated dose, and so it could well be that there are unwanted, we call them side effects, but toxicity of drugs, because every drug is toxic. If you take 2 aspirins, perfectly fine. You take 1,000 aspirin, it's not so good. So the dose matters, and unhappily, back to the question, takes time to determine where it is, and so that will be a 2 to 3 year outcome to recruit enough patients to the full cohort of patients, and then see what the effectiveness or absence of effectiveness of the approach really is. Yeah.
But wouldn't that be
No.
Have a repository of information of people who are going into these clinical trials, where you have all these blood biomarkers and other biomarkers say they respond, they don't respond. I mean, it's simple, right?
Yeah.
I mean, people going into clinical trials.
Yeah.
You're just going to take people randomly off the street, or people who have some symptoms.
So the question about trial entry, and yes, there are. I mean, that's basically it's trial design, and I think it's actually a very it's possible retrospectively to recognize that you designed it wrong. It's easy to criticize the trial design. The SOD1 trial was run too short, obviously too short. I'm sorry, it was, they weren't really, that was a clear design error, prospective design error, but okay, yeah, yep.
Thank you so much for this session and for your time and energy and
Yeah, thank
answering
you.
all questions. Why don't we give a round of applause to both Dr. Senda and Dr. Senda. Thank you so much. So it is also my honor to invite Laura Fravelletti to make the closing remarks for the meeting. And I would like to tell you that there are many people online watching this event, and we have more than 100 people joining all around the world. They may not be here in this room, but your voice is actually heard. You know, from Europe, from South Africa, Japan, we have people joining at night, in the morning, at different time zones. And what we say here really matters. It echoes. And I'm very happy for all the presentations, for the patients, their voice, their input. And I'm also very thankful to Laura Fravelletti for her closing remarks. Thank you, Laura.
Thank you. Thank you, Hyundai. And don't worry, I really don't have closing remarks, but what I have is a big thank you to everyone who's here, everyone who has stayed, especially to our expert speakers. Very, very appreciative of your time today. I would also just add that there are some things that are abundantly clear today that we heard. One is that progress in ALS requires partnership, and it's wonderful to see and hear the partnership that is going on. Second is this work requires sustained philanthropy and support. And finally, two words that are always near and dear to me. One is resilience, that we see from people living with ALS. And the second is a word that Dr. Cleveland used, which is perseverance. And that is the work that all of you scientists do. So thank you again very much, and look forward to seeing you next year.